In this lecture, we are going to discuss the different types of agglutination methods. Well, what is agglutination? Agglutination is the precipitation and agglutination that are visible result of antigens and antibodies aggregating together. Okay, so if you were in the immunology lab this week, you may have noticed that we did a CRP agglutination test where you could visibly see some of these happening. We're going to talk about a couple different types of agglutination that are very similar but not exactly the same. So precipitation is the formation of a solid mass from a previously soluble component, a soluble antigen and a soluble antibody. Something that uses this is in RPR test that we'll be doing later on. That was one example of a precipitation type of test. Agglutination is the visible clumping or aggregation of particles that have antigens on their surface by antibodies that form bridges between the antigenic determinants. Examples of some artificial carriers that we could use to make this happen include latex particles and colloidal charcoal. You'll be seeing both of those used in the immunology laboratories. Some natural carriers include cells, and I'll show you some pictures of that in a little bit. Another one is co-agglutination. This uses antibodies bound to a particle to enhance visibility of agglutination. So how does latex agglutination exactly work? Well, antibodies are actually bound to little latex beads. If the specific antigen is present, it'll bind to the sites on the antibody exposed on the surface of the latex bead and form an aggregate. You can see this happening here. We've got the antibody stuck to the beads, okay? We add the antigen, they combine, and we get this whole lattice formation here. Here's an example. We can do it on a card. Okay, so card agglutination would be clumpy, like you see on the left, where negative would be creamy and homogeneous. In a test tube, agglutination is going to be clumpy, where non-agglutination is going to be relatively smooth. So we do have some phenomenons called prozone and postzone. Prozone occurs when there's way too much antibody. There are two antibodies bound to every bivalent antigen and no need to bridge two antigen molecules by an antibody. There'd be little or no precipitate that would form. We always think of this as like pro football players or pro athletes have good bodies, so we put the prozone and the too much antibody together. Then we can have postzone. This can occur when antibody is too low. So each antibody is bound by two antigens and there's no bridging between them. No precipitation occurs. So if you have a situation where there's so much antigen present, some disease scenarios or something like that, where the antibodies are just overwhelmed, we would call that, postzone. Prozone is where there's too much antibody. Postzone, too little. Okay. Zone of equivalence, the ratio is just right. Okay, that kind of makes sense because it sounds like what it does. Next, we have flocculation tests. These are based on the interaction of soluble antigen with an antibody that results in a precipitate of very fine particles. Sometimes they're macroscopically visible, sometimes we have to use a microscope. A couple different types of testing for this. The Venereal Disease Research Laboratory, which tests for syphilis in spinal fluid, and the rapid plasma reagin, which is used to test for syphilis in the serum. We also have direct bacterial agglutination. This is where pathogens are used to detect antibodies directed against pathogens. So for example, if they're in the microbiology department and they have some bacteria growing and they're not sure if it's a staph or a strep, they can use staph-coated or latex antibodies, latex and antibodies stuck together and add the staph or strep that they are not sure what it is. And if it does agglutinate, then you know that it would be staph that's present because that's the antibody connected to the latex. We also have indirect or passive hemagglutination. Hemagglutination is where they use RBCs. So this tests for antibody detection. The first thing that happens is RBCs are coated with a substance, usually bacterial cells, fungi, or protozoa. And it's called indirect or passive because it isn't the antigen found in the RBCs, but the coated substance that the antibody will bind to. In other words, the antigen passively attached to the red blood cells are bound by antibodies. So hemagglutination detects those antibodies to RBC antigens. You can see the specimen that it contains the antibodies looking for will be diluted, and RBCs are added, and it will cause the agglutination. We also have agglutination inhibition. This is where agglutination represents a negative reaction and no agglutination represents a positive reaction. So it's the opposite of what you would think. We do have different factors that influence agglutination. First, there's two stages. The first stage is sensitization. This is the physical attachment of an antibody to an antigen. Okay. From there, we have lattice formation. These are the crosslinks that form between sensitized particles. So we have antigen, antibody, antigen, antibody, antigen, antibody. They're all kind of stuck together. The ability of the cell with the attached antibody to come close enough to another cell is what we're referring to. The reason why this is so important is because we have to overcome zeta potential. Zeta potential is the degree of repulsion between cell membranes. All of your red blood cells floating in your bloodstream have a slight negative charge to them, okay? And that can become a problem when we're using hemagglutination, okay? So we need to find a way to force them together. Here are some ways we can do that. Centrifugation, this attempts to overcome distance between sensitized cells by a high gravitational force. We can treat them with enzymes that alters the zeta potential by stripping off some of those negative charges I just mentioned. We can use colloids, which will adjust that zeta potential. And we can actually use anti-human globulins too, which are antibodies that don't react with the RBCs, but they bind with the antibody on it instead. When we look at agglutination in the laboratory, we have to give it a grade, okay? So when there's no aggregation present or no clumping at all, we give it a grade of a 0. That means there's no aggregates. A mixed field, there's few isolated aggregates, mostly free-floating cells. Your supernatant still looks red, so the sample still looks red, if we're dealing with blood cells, that is. A weak positive is where there's little tiny aggregates, barely visible, hard to see. 1+, there's a few aggregates that you can see, but there's still pretty red supernatant. 2+ is where we start to see the clear supernatant, okay? That's where we see some medium-sized aggregates. The back or the serum would be clear. 3+, several large aggregates, and 4+ is one solid aggregate clump. Here's what that's going to look like. I know it's kind of an archaic picture, but it's a good one. So when you have a 4+, you're going to have a big clump of cells, okay? 3+, you're going to see some large aggregates here. 2+, you're going to kind of see a cloudy background, but you're going to have a lot of little clumps going on. 1+, very cloudy background. Negative, it's all homogeneous. There are some other issues we can have. Pseudoagglutination, this is the false appearance of clumping. It can look like it's clumped, but it could be due to Rouleaux formation instead. Rouleaux is usually found in patients with a higher abnormal type of globulin in their blood. On a microscopic exam, the RBCs kind of look like stacks of coins. Usually we can add a few drops of saline to resolve it, but this is kind of what it looks like. And it's a very common cause of pseudoagglutination, and you can see it looks like a stack of Rouleaux, if you've ever seen like that candy. Rouleaux and Rouleaux sound very similar. And that is the end of our agglutination presentation.